Okay, hi. So this is going to be a revision video for B3, which has 3 parts. So it has the gas exchange, the transport, and then the movement. So we'll start with a gas exchange. And throughout this topic, remember we are looking at it in animals and then in plants within each subtopic. So for your first page, for your one on gas exchange, leave a space at the bottom for plants. So make sure to leave maybe at least a quarter of the page or so. And then we'll do plants afterwards. So we'll do all things animal. This topic is fairly SL heavy as well. So there's really only one little part that'll be HL, which is going to be the stuff on hemoglobin. The rest of it is all everybody. So just to kind of let you know about that, most of this page will be for everyone to be writing. So let's start with in animals. So we'll go first into in animals. And then go to plants next. Okay, so I want to start kind of big and then go small. So I want to start with ventilation. So big picture gas exchange, and then zoom in to what's actually going on in terms of gas exchange in the alveoli. And then we'll zoom in even more for higher level and look at what's going on with gas exchange at the actual hemoglobin level. So going from big to small. So in doing that, let's start with a definition of ventilation. So ventilation is going to be the exchange of gases with the environment. So that is between the organism and the environment. So it's in and out. So the common term for that, of course, is breathing. So ventilation is just your fancy term for exchanging with the environment. So exchange of oxygen and carbon dioxide with the environment. So quickly, let's look at why is ventilation important. So it should be pretty obvious, but why is a better way of saying it, maybe why is frequent ventilation important? So we do breathe about every 10 times a minute, so every 6 seconds or so. So we do breathe fairly often. So why do we have this kind of frequent exchange? And the reason for that is that we have to maintain concentration gradients. So we can't just breathe in oxygen and then just wait until we use up all of that oxygen before we exhale. So that would actually take a couple minutes if that were the case. So we would only be breathing every 2 or 3 minutes, which is why you can hold your breath and survive for longer without breathing. But if we do that, we slow down the rate of transfer because the oxygen hits equilibrium and then it doesn't really move very much after that, and you would have to wait for it to drop in the blood due to respiration, and it would dramatically slow down our body. So the reason why we do ventilation all the time, the reason why it is such a frequent thing, is because we are maintaining the concentration gradients, because the goal is going to be for gases to exchange passively and not be using energy for that. So we should know this term well by now, but the idea is we're maintaining the concentration gradient that we need so that there's always more oxygen in the lungs than in the blood that's passing through it. So maintain that concentration gradients for diffusion from high to low. So the key is we need to ensure that we are keeping a situation where there is always high oxygen in the lungs. So that is why we breathe frequently. Okay, so let's look at what this looks like. Our main focus will be on mammals, but there's a bit of a fun theme throughout all these topics of like, oh, but what about the fish? So we will quickly look at what the fish do differently, but we're gonna mainly be looking at mammalian systems. So I'm going to do a little mini, I'm not going to do a full respiratory system diagram. We'll kind of cover some of the key parts in other things. But I'm going to do like a little mini kind of rib cage area. Because when we talk about ventilation, we are talking about inhalation and exhalation. And you do need to know what's happening to facilitate that. So I'm going to draw a little trachea. Do you need to know that term? Then I'm going to draw some ribs. Like so, and then I'm going to add some more things on. So surrounding the ribs, of course, is the actual lungs. and how big the lungs are. Maybe I should be a little bit closer to my actual ribs, but that's all right. And then there's 3 muscles that you need to know about. So we're gonna have it sitting down here is gonna be the diaphragm. And then we're gonna draw 2 different intercostal muscles. So we're gonna do the external intercostals, which are gonna sit kind of on the outside of the ribs. So they're kind of more like that. So they're hitting just across the outside of both ribs. And then we're also gonna draw some internal intercostals as well, which are linking on the inside. So you do need to know about those different muscle groups. Okay, so let's go through what's happening for both inhalation and exhalation with each of those different things. So we're gonna make a little mini chart, and then we are gonna use the unnecessary IB terms of inspiration and expiration, just so that we remember that those mean the same thing. So inspiration is inhalation, and expiration is exhalation. So means the same things. So for each of these, the things that I would want you to know are what's happening with the diaphragm. So what is the diaphragm doing at this moment? I'm just writing it in the same color that I put it in the diagram. Next thing I would want you to know is what's going on with the intercostals. So what's going on with the intercostal muscles? So intercostals. And there are 2 sets. So I'll leave a little bit more space for that. And then I want us to be able to say, what does that do to the volume? And then what does that do to the pressure? So then the question becomes, what happens then to the lung volume? And then finally, what does that do to the lung pressure? So the inverse relationship between those 2, it's called Boyle's law. I don't think you need to know the law by name, but do need to know it in this specific context. Okay, so let's go through what's happening for each of those 2 processes. So when inspiration is happening, the diaphragm contracts. So the diaphragm is contracting. And when it contracts, it's going to pull down. So that is going to pull the diaphragm down. Like so. Okay. At the same time, so that's going to make more space, we also want to expand the ribs out. So in order for that to happen, the external intercostals are going to contract. And the internal are going to relax. So my internal relax. That actually makes them stretch. And so by stretching those, you're going to actually push your ribs out. So that is going to push the ribs out. Remember, this actually comes up twice. So they ask about them as an antagonistic pair again under motility if you're HL. So if you're HL, you definitely want to make sure you're good with your internal and external intercostals because they do come up twice. As a result of the diaphragm down and the ribs out, the lung volume is going to increase. And then, like we said, because of the inverse relationship according to Boyle's law, this pressure, therefore, is going to decrease. And then because of the low pressure, the gases are going to move in. So the low pressure is going to cause the gases to move in. So they're going to follow that pressure change. So they're going to move in from the environment. Okay, so then let's look at the opposite. So when you exhale, the diaphragm is going to relax. And when it relaxes, it's going to pull up. Bit tricky here because it depends on the how kind of aggressively we exhale. So at all times, the externals are going to relax. So your externals relax. And as they relax, that is automatically going to pull the internals in a bit. So as they relax, they let go of that stretch. And so the internals can contract. So they don't actually need to. they can just release a little bit of the tighten pull and kind of come back to shape, and that's enough for a normal exhale. But if we're trying to do a purposeful exhale, we will have to use extra force from our internal intercostals. As a result, because this is going to pull the ribs back in, the ribs pulling in is going to reduce the volume available for gases. That is therefore going to increase the pressure they're under, and then as a result, the gases are going to be pushed out. So that is going to force the gases to exit. Okay, a couple more things on ventilation. So the other kind of two things I want to look at is looking at kind of tidal volume versus vital capacity. So being able to make sense of that information. And then we'll do our quick, what about the fishies moment? So while we're still on big picture stuff, let's look at lung volume. Because we recognize here that lung volume is important. So we know that the ability of the lungs to expand and increase in volume is important for being able to take really good deep breaths. So for this, we're going to draw 4 lines. Will work. Like so. Okay, so you can get this from like a spirometer, so it is possible to kind of gather this data. Of course, you're not doing prac work in your exam, so they would have to give you the data. So for the key for you is going to be to interpret it. So we actually use a small portion of our lungs normally. If we look here, all of this requires muscle contraction, therefore requires ATP. So we do not want to exert a ton of energy just to breathe. So if we're using big contractions and relaxed things, we're constantly using a lot of muscle energy to move air. We don't need that because, again, it's better to do it more frequently. So smaller but relatively frequent breaths is the best way to maintain concentration gradients. So we normally actually use a small amount of our lung volume. If you take a really big inhale though, there's a lot of extra room that you could use. And then if you do that, you're going to release a big exhale. But we will remember, you don't need to know the name of it, but we don't actually use, there's a tiny reserve volume that we try not to fully exhale, so that if we have an emergency, we have this kind of adaptation to hold a little bit. The main thing is just going to be to know the terms for these. So this that we normally use is called the tidal volume. So that is the day-to-day lung capacity that you are using. This kind of extra ability to inhale is the inspiratory reserve volume. And then this extra bit that we could exhale, if we've inhaled big, is your expiratory reserve volume. And then this whole area that is able to be consciously used when we take a deep breath is your vital capacity. And these are both really important. So ideally you want to have a good vital capacity, and you also want to use a fairly small tidal volume. So you are best adapted if you're running on a small amount, but have a big amount you could use when needed. Okay, so like I said, this is all things mammals. Just really quickly, how does this work in organisms that use gills? So their method of kind of exchange is a little bit different because they don't have lungs. So they have a particular adaptation called counter-current exchange. So counter-current meaning currents, movements in different directions. So they inhale or they take in dissolved oxygen, so a very different thing because it's already in a liquid form. So that's kind of why it's a little bit different. So here's let's give them some gills. The gills are the actual slits. These are like the tissues in under, which I've just drawn a blank, like Laird slits or something. You don't need to know that name. If you're a fish person, sorry, I'm not. Okay, so then they are going to take in dissolved oxygen. So this is going to be oxygenated water. And again, the word current, because it's water, is then going to move across the different gill slits. So those different areas that are capillary heavy for exchange. And then as the blood goes through this same area, it's moving in this direction. And it's essential that at every point, the amount of oxygen is always higher. So at every point, the oxygen is higher in the water than it is in the blood. So as it reaches the end and it's running low on oxygen, because that is coming into account with the lowest oxygen blood, it can still move it. So if they were running at the same direction, it would hit equilibrium early on, but by running in opposite directions, they're able to maintain it at every point. So it is always going to be higher oxygen at every point. At every gill. Not sure I'm using the word gill completely accurately, but you get the idea, the gill strips on the inside. Okay, so that is countercurrent exchange. It's just an adaptation that allows them to use dissolved oxygen in a way that maintains concentration gradients. Okay, so that is all of our big picture stuff. So we're zoomed out ventilation. We're now going to move into the kind of more small scale stuff in animals. So the actual exchange of the gases within the body. So different terms, you can see this called external respiration, and then internal is at the tissue level. IB does not clarify those terms, so we don't need to worry about those too much. These are both just gas exchanges in the body. So we're going to look at gas exchange at the alveoli. And then if you're HL, we're then on the other side of this going to look at the actual gas exchange from hemoglobin. So leave space for that. If you're SL, you won't need that part. So you can draw a really giant alveoli if you want. It would be strange because they are in fact very small, but if you so desire, draw very large. Okay, so the other part of the respiratory system, we already did the trachea. The other one that you do need to know is this is a bronchiole, which branches off from the bronchi. So we have the big bronchi coming straight off the trachea into the smaller tubes called the bronchioles. Okay, so I'm not going to draw tiny, tiny little cells. We did that in class, so you can go back to that one. Kind of makes it slightly more crowded looking. So we're just going to label it as one cell thick, but not worry about drawing to show that each of these is a cell. I am going to draw a few type 2 pneumocytes, just to clarify that there are some of those there, and then we'll assume everything else is that kind of one cell thick. So I can kind of draw one just to show you what I mean. So we could line it like that, but we'll leave that off just to make it a little bit simpler. Okay, so there is one alveolus. So alveolus is singular, alveoli is plural. So remember, in the lungs are filled with lots of these, so they are tiny. Okay, so and then on the inside with the gases, the easiest way for them to move into the blood is to go ahead and have them dissolve. So there is a thin layer of moisture on the wall. So that's going to be moist for gases to dissolve. The problem is that these alveoli are so small that if you had the moisture and nothing there, then when you exhale and they lose the air, the cohesion between those water molecules would suck the whole alveolus into a little shriveled up thing that would stick together. So to prevent the cohesion, we are going to have, so these, remember, are making, so they're releasing a bunch of surfactant. That is their job. And surfactant is like a single little phospholipid molecule. So it has these little fatty acid tails. So instead of a bilayer, it's actually deliberately like this, whereby the polar head sticks to the water and then the tails are sticking out. So that is going to be my surfactant. And remember, that is made by the type 2 alveoli, and that is going to prevent collapse And it does that by preventing cohesion. So preventing the lung collapse by not allowing the water to cohese during exhale in particular. So that's going to be really important. Okay, let's also make a note that this wall is 1 cell thick. And that's going to be really important so that we have short diffusion distances. So we have to be a little bit careful when you talk about the adaptations. So when we talk about 1 cell thick, the thinness, it's about the distance. If we're talking about how many small alveoli there are, that's the surface area and the speed. So let's clarify that as well. So we're going to say that the lungs contain many very small alveoli. And the reason why that is important is when you have many, that is going to increase your surface area to volume ratio, and that is going to increase the speed of the diffusion. So it's just able to move more quickly if it's happening in more places. So faster versus the short distances, which are related, of course. The short distance will make it faster, but not specifically because of the surface area. So just kind of be a little bit cautious about that. Okay, so that's all things going on. And of course, if we want the exchange, then this needs to actually be covered by a capillary. So here's my capillary. My capillary is also 1 cell thick. So again, I could draw this like that and draw that. But if I do that, then again, it's gonna look super crowded. So I'm just gonna label at this point that the capillary is also thin. And note that it is 1 cell thick. So again, between those two things, it is going to ensure that it has the short distances. So my blood is going to come in from a vein. So coming off of, it's going to come in from a pulmonary artery. So because it's coming away from the heart, it is going to release its CO2, which will ultimately then go out and go back to the lungs. And then in is going to come the O2, and that is going to go into the cells, and they are now oxygenated, and they will go back to the heart and the pulmonary veins, and then they will be pumped through arteries of the body. So that is where the actual gas exchange happens between the lungs and the blood. Okay, so I think that's all things you need if you're SL. Just chill for a minute, fast forward a couple minutes until you start seeing me draw a plant. So when you start seeing plants, come back to us. If you're HL, we need to do a little bit more because we need to actually talk about how that exchange happens at the hemoglobin. So we are going to look at the hemoglobin and the Bohr shift. So, like I said, we could be calling that external respiration. And then this would be internal respiration. So essentially we need to exchange it between the blood and the lungs. And then we also need to exchange it between the blood and the cells. So this is the next zoomed in level. For simplicity's sake, I'm going to, I don't know if simplicity is the right word, but I'm going to draw all of this with 3 shifts on one single graph. So use kind of your space for this to draw a reasonably sized graph because we're going to have to write things on it. Okay, so what we are drawing here is the oxygen dissociation curve. So this is an image of that is giving indication of when oxygen is associated and when it dissociates. So dissociates is looking at when does it fall off the hemoglobin. So this is looking at whether oxygen is associated with hemoglobin or comes off the hemoglobin. And remembering that when it comes off the hemoglobin, it's going to the tissues. So that is a beneficial thing. Okay, let's remember what we label this as. So this axis is going to be the percent saturation of hemoglobin. You can use HB for hemoglobin. That way I don't have to stress about whether I remember to put the A in or not. And then down here, we're going to do the partial pressure of oxygen, which is basically just another way of saying the concentration or the amount. So how much oxygen is there? So the partial pressure is the amount of the gases that is oxygen. So that's just kind of an indication of that. There. Squeeze it over a little bit. Okay, so let's draw the traditional curve and explain it. So again, if there were not cooperative binding, what we would see is it would just continue to increase. So as you increase the available oxygen, more of it binds to the hemoglobin. It does not work that way. So it kind of very quickly starts out. It's actually reasonably hard to bind. And then as you get kind of 2 on, and then you reach that point where it goes dramatically up. So it's going to end up looking kind of like a sigmoid curve. So kind of slower and then basically vertical at a point. So when you hit about the 50% saturation, that means there's generally 2 oxygens. The third one is super attracted, and the fourth one is so attracted it basically goes from 75 to 100 almost immediately. Okay, so we're showing kind of that sigmoid curve, and then we want to make a note that the reason for that curve is because of cooperative binding. So each bound hemoglobin increases the affinity for other ones. So this is my non-linear curve, and that is due to cooperative binding. You do need to know that term and what we mean by it. So when we say cooperative binding, we are saying that each bound hemoglobin increases affinity. So we'll just kind of quickly note that so that we don't forget. So each O2 that binds to hemoglobin increases the affinity for the others. So the easiest one to bind is the fourth one. So you're basically going to go from saturation point of 75 to 100 super quickly. That's really valuable because it essentially means with a relatively small increase in oxygen, you can saturate your hemoglobin. So that means with ventilation, we can increase our oxygen concentrations, relatively small amounts, boom, saturate our hemoglobin. So just by inhaling fresh oxygen from the environment that is not 100%, we can quickly saturate the hemoglobin. Okay, and then it's equally worthwhile noting that in all cases, that at low oxygen concentrations, the oxygen will dissociate. So in all of these situations, and we're going to go to the even more dramatic version of that in just a second, but in all situations when there is low oxygen, it actually causes the oxygen to lose its attraction to the hemoglobin and it will come off. So low oxygen, we lose kind of oxygen, so we meet the needs of the respiring tissues. On that note, let's continue to say, okay, this would just be under normal circumstances, but we have an adaptation that is extra helpful. That means not only does the amount of oxygen matter, but the actual amount of carbon dioxide matters. And what that does is it allows us to preferentially lose more oxygen at tissues that need it more versus a kind of holistic overall kind of unloading. So what happens is if the carbon dioxide levels go up, the pH goes down. That does 2 things. The general pH being lower causes that losing of the bonds, but also allosteric bonding. If you have more CO2, more of it hops onto the hemoglobin, which causes the oxygen to drop off. So in the presence of CO2, you see the same general curve, but more dramatic. So it will basically look like this. So it won't really matter at the very highest, but what it's showing me is that you can start losing oxygen earlier. So even if there's still decent amount of oxygen, like in the overall area, if you have high CO2, you will drop the affinity quicker. And so it's a sensitivity towards high CO2, representing the idea that there's a lot of cellular respiration in that case. So this trend, this movement out, is called the Bohr shift. And the Bohr shift just says, at lower pHs, which is due to high CO2 production, the affinity for oxygen decreases. So you have a lower affinity at every point. So essentially, your affinity is going to increase more rapidly. So again, this is due to the pH, but also, let's make a note, due to allosteric binding. So it's going to hop on, not to the actual oxygen binding spots, but it's just going to hop on to another place, changes the shape, and then it causes the oxygen to fall off. So due to both the pH change and that's going to happen. The benefit of this is that when you have high CO2 and that creates a decreased affinity, this is helpful because what it means is it means that oxygen is delivered to tissues that have high CO2 or tissues that are really actively respiring. So rather than waiting till their oxygen gets low, the fact that they have low CO2 or high CO2 suggests high rate of respiration, so we can deliver oxygen quicker to them. So give them extra oxygen. So it's a really beneficial thing that our body is sensitive not just to oxygen amounts, but to CO2 amounts, and it helps us kind of get the right amount of oxygen to where we need. I'm just gonna make a note of this. I forgot to do this before. This is our HL only part. Okay, the last little hemoglobin application that you need to know. So this one is going to shift it to the right. So we are going to see a lower affinity. We also want to draw the curve for fetal hemoglobin. And so fetal hemoglobin is going to be to the left, meaning that it has a higher affinity. So this would be fetal hemoglobin. So it has a structural difference. So it has Y chains instead of beta chains. And those structural differences change the binding, how tight it binds. And therefore it changes its affinity. So this is going to have an increased affinity for oxygen. So it is going to pull oxygen more. So at every basic place, no matter what the oxygen saturation or concentration is, the hemoglobin is going to prefer to go to the fetus. And that is going to be important because it allows for oxygen transfer from the mom at the placenta. So when it's in the placenta and the mom's blood and the baby's blood are in close proximity, the oxygen has a higher affinity or attraction to the fetal hemoglobin, so it will pop off the mom's hemoglobin and go to the fetal hemoglobin, which is how we can meet the needs of fetuses that do not have the ability to do their own gas exchange. So they are reliant on this process. Okay, so that is all things the Bohr shift. Just make sure you do understand kind of cooperative and allosteric binding. I didn't go into a lot of detail. You don't really need to know what the shape change is, although it might be worth just reminding yourself that in both of these cases, it is related to the shape change. Just because this is the kind of form and function unit, so you always want to be able to link the structure. So the structure changes, therefore its affinity changes. Okay, so that is all things in animals. We're going to do a little bit on in plants to wrap up gas exchange. Plant stuff is everyone for this topic. So this is SL and HL. So we all have to know the same things. So gas exchange in plants. And just like within animals, there's this overlap because we're talking gas exchange. We have to talk about the capillaries because that's kind of where it happens at. And you have to talk about hemoglobin, which is in the blood system. So similarly, we're going to talk about gas exchange, which is closely linked to transpiration because technically it is a loss of water vapor, but then transpiration is also important for transport. So we'll kind of come back to it kind of later as well. Okay, so we are going to draw a little leaf cross section here. So we need to draw some cuticle, particularly on the upper side. We might have a bit of cuticle down here as well. The cuticle itself is made by the epidermal cells. That is all you really need to know about them, is that they make cuticle. The cuticle itself is the waxy kind of waterproof layer. So this is going to be waxy and waterproof. So it should trap the water in. So this is like covering something in Glad wrap. Should be trapping the moisture in is the goal. And then we will have this as well on the under surface. Okay, then we're going to have two main types of cells, which are going to be your palisade mesophyll and your spongy mesophyll. So your palisade are going to be your long cells, like so. Importantly, they are long and thin on purpose so that you can get more cells across. So you have more cells that are close to the surface, close to the sun. So they are going to be long and thin palisade mesophyll cells. So lots of them packed in. The other thing we need to indicate is that they have lots of chloroplasts. They generally have lots of chloroplasts around the outer ring just because of the fact, and I'm not doing a very good job drawing chloroplasts, but we can just label them chloroplasts. We're not drawing them straight through the center because they have relatively large central vacuoles because they are going to want to maintain enough water to do photosynthesis, because remember, water is a reactant of photosynthesis. So I will kind of label, in case we're not clear, that those are chloroplasts. And these are going to be the most heavily photosynthesizing area. So these are going to be doing lots and lots of photosynthesis. So they're on the leaves, close to the surface, near the sun, with lots and lots of chloroplasts. We're going to have some more cells here, but we need to have some space. If we chocked it blocked full of palisade mesophyll, the gases would have a hard time getting into each cell. So we essentially need each cell to have some space where gases can filter in and diffuse in. So we're going to have some more kind of cells that will also help with photosynthesis, but they're going to be spread out. So this is the spongy mesophyll. And the idea will be that the gases can travel through that area. So before I forget though, let's also just quickly draw some xylem and phloem. So we need to also deliver water to actually be able to photosynthesize, and we need to be able to collect the actual glucose after we make it, which will happen in the phloem. So that is going to be a vascular bundle. Because we are going to talk xylem and phloem in the next section, I'm not going to differentiate them and talk about their function here. I'm just going to label that. Okay, but that is going to provide the other things that are needed. So there's my spongy mesophyll. What I actually need is I need to actually have an area where they can actually come in. So then I am going to have this gap, and the gap is where the oxygen is going to come in, and where, the carbon dioxide is going to come in and the oxygen is going to go out. That is the stoma or the stomata. That is determined by the guard cells. So if the guard cells are nice and full of water, so here's some nice turgid guard cells. So these are turgid guard cells. So they are well hydrated. Then they will generally be open. So this means the stoma is open. However, if they start to lose water, the water will start to come out of the vacuoles and the cells themselves will become flaccid and they will end up moving towards each other. So as they become flaccid, they actually move towards one another, and these are going to be flaccid guard cells. And they are going to have no gas exchange possible. There are other things that can close guard cells. So you can also have just hormone chemical messages that tell it to close its guard cells. So it doesn't have to only be due to water loss. They will often close at nighttime just because there's no photosynthesis. So one thing that can close them is the loss of water. There are other things as well. Okay, so while we're on gas exchange. So gas exchange, this availability of gas exchange requires the open stomata. When the stomata is open, transpiration is also happening. So it causes transpiration. So if we recall, transpiration is the loss of water vapor. So it is the loss of water vapor. So it's already evaporated. So evaporation has to occur before transpiration, and the water vapor is lost from the open stomata. So loss of water vapor out of the open stomata. The other thing you need to know is what increases and what decreases it. So in what case is this increased? And then there's one thing that decreases it. So 3 things that increase, 1 thing that decreased it. So the first thing that increased it is going to be increased sunlight. So in the presence of increased sunlight, the cell recognizes the sunlight and wants to do more photosynthesis. So that is going to increase the gas exchange here. And the reason for that is in order to increase photosynthesis. So because of the need to pull in more carbon dioxide, the stomatas are going to stay open for longer. Okay, next one is next thing that increases it is increased temperature. So you'll notice I made a point up here that is the vapor that leaves. So if you have an increase in temperature, you have increase in evaporation. So that means there is more water vapor available, and that is due to the increase in kinetic motion. So essentially water is more active. So that causes more evaporation as well as just kind of faster movement even through the xylem. So in general, the increased kind of kinetic motion of the water pulls more of it up, evaporates more, and therefore produces more vapor to leave. And then the last thing that we also need to understand that can increase this is wind. So what wind is going to do is it's going to blow the recently released water away. So it's going to remove water near stomata. And when you do that, that is going to make the air drier. And that is going to essentially decrease the water potential of the air. Or if your SL, increase the concentration gradient. So the difference in concentrations. So either one. So increase the difference in the concentrations. If you're HL, you can use water potential. If you're SL, just think of it as the drier it is, the bigger the difference. So therefore, the more water is going to leave. The only factor that is going to reliably increase it is going to be humidity. So as we increase humidity, that's going to do the opposite of this. So there's going to be increased water in the air. if we have increased water in the air, that is going to increase the water potential of the air. So that is going to increase that, which means there's going to be less of a difference. So that's going to reduce the difference or the concentration gradient. So generally speaking, the air should have low water potential, but if we actually have a lot of humidity in it, it will not have as low of water potential. Okay, so that is all things all things gas exchange. Next up is transport. Okay, we're on to transport now. So essentially, I did actually draw lines on mine, but we're gonna use the bottom kind of third of the page for plant stuff. So leave the bottom. You might want to just, when I did mine, I just went ahead and drew a line down there so I didn't forget about the plants. So the bottom is going to be for plants, and then the top kind of 2/3 is going to be for all things animals, and then it's basically going to divide in half again if you're HL. So if you're HL, we're going to put the SL stuff related to vessels on this side, and then we're going to put the heart stuff on this side. If you are SL, you have the whole space for the vessels. So you may use the entire top on this next section. If you're HL, leave some room. Okay, so this is a kind of interesting division in terms of what they decided was HL and SL. So SLs need to know quite a bit about the blood vessels, but you don't really have to know the heart at all, which is very tragic. And then HLs, you need to know blood vessels, and then what we're adding is the heart stuff. So we'll start with the vessels since they are everybody material. So blood vessels. And again, I'm just going to even, for my own self, remind myself that I'm going to stay in my lane because all of that is going to be the HL stuff next. Okay, so we have 3 big vessels, and then you do need to know kind of the names of the connecting vessels as well. So we are going to have the arteries. And then the arteries are going to branch off into a capillary bed. And what makes it a capillary bed is that these should all be coming off of the same arteriole. So arteriole is just the smaller vessels coming off of the artery, and then the whole capillary bed will be attached to that. This is zoomed in on one cell, so remember they are not the size, so this is super zoomed in. And then they will all then connect back together via one venule, and then we will have the veins. I'm going to also, this is HL only. I know I'm not staying in the right side, but it makes more sense. So you have to know one other vessel, which is going to be lymph capillary. So I'm going to draw a little lymph capillary as well, and just do the little lymph part right here because this is where it makes sense. But this is HL only. So if you're SL, they don't exist. You have no lymphatic system. Okay, so let's have a look at the kind of structure to function. So again, that is our emphasis. So what do they look like and how does that help with their function? Okay, so with our artery, we need to have a very normal, they both have the kind of tunica outer. You don't need to know those names. Then they're going to have that really big tunica media, and then they have the tunica interna. But the main thing is this really thick inner wall. That thick inner wall contains smooth muscle, which is involuntary muscle. It contains elastic fibers, and it also contains collagen, which is just kind of a tough protein that will give it a kind of more durable, sturdy strength to it as well. So those are the things that you need to know. I always think it's good to come back to what is the adaptations for. So for arteries, these are adaptations needed to withstand high pressure. And the reason why it's going to be high pressure is because all arteries are moving away from the heart. So it has always just been pumped. So because it is moving away from the heart, it has lots and lots of force because it's been recently pumped. So the three things that we kind of drew here, we're just going to clarify the three adaptations for that. Is this really thick wall, including the fact that it has that collagen in it. As a result of the wall being so thick, the inside or the lumen is very thin. That's going to be important because it allows for it to be secure, and it also protects the blood in the arteries because of the force. It's also really important that we don't damage our arteries. So it has kind of two purposes there. The benefit of the muscle is the muscle allows the wall to move in and out. So the muscle will adjust the wall. So it is going to enable it to essentially stretch out when there is high pressure and then move back in. So it is going to cause that kind of adjusting. So it's going to adjust the wall and consequently the size of the lumen. And then the other one that they've added in that you need to know is the elasticity. And what they actually do is they help with movement. So they actually add to the movement. And they do that via their stored potential energy. So when they pull out, they store some potential energy. So they help push the blood forward and help it propel. Okay, so those are all things that are adaptations of the arteries. So then we come from the arteries to this arteriole. And then the main goal here, so we said here for the arteries is all about high pressure. The main goal here needs to be fast diffusion. So this is where the actual exchange is going to occur, and we need to have efficient diffusion of substances. Okay, so a couple of adaptations we need to note for that. First of all, we've drawn it as one big one, but let's make sure we know that there are in fact many very thin vessels. And again, because we have many thin vessels, that is going to increase our surface area to volume ratio. So that is going to speed up the diffusion, which is really important. And remember, when we say thin, they are essentially, we haven't drawn it this way, but they are 1 cell thick. Which again is that idea of the short distances. So if I was actually drawing it, this whole thing is 1 cell. So not just one layer, but at the absolutely capillary, the entire vessel is only the thickness of that kind of 1 cell stretched across. In terms of how thin they are as well, 1 red blood cell can fit like this in it. So that shows you how small that is. That is a single red blood cell along. The next thing we need to show, which I didn't really show here, so let's just, whiteboard markers for the win. They have little fenestrations, so that is a fenestration. Not all capillaries have them, but a lot of them do that are in kind of high exchange areas like the intestines and the kidneys. So the benefits of the fenestrations is that they are going to allow for more things to move through it. So fenestrations are going to increase the permeability, so more things can get in and out. Perfect. On that note, this is HL, when you are actually going from an arteriole to a capillary, there's going to be a big change in pressure because it's going into a really small vessel suddenly. And between that and the fenestrations, you are going to lose tissue fluid. So tissue fluid is going to be coming out. The tissue fluid is going to be really important because that is where the actual exchange is going to happen for most cells, because most cells are not right by a capillary. So the tissue fluid is going to allow them a little bit further movement. Sorry, I'll write that in a color that's actually visible. So the tissue fluid is going to come out. And then the tissue fluid is going to try to go back in, kind of at the arteriole, or the venule, sorry. So we'll have kind of a return of tissue fluid there. However, a lot of tissue fluid does not actually get returned to the venule. Even though there is reduced pressure allowing some of it to come in, it's not enough to bring it all in. So the rest is going to actually be absorbed by the lymph capillary kind of later. So it can actually slowly diffuse into the lymph capillary. So the lymph capillary is going to collect it, and then ultimately it'll filter it back into the veins. So it'll connect it back. Okay, so that's kind of another little tissue to know about. So let's just, again, for HL, only clarify what this is doing. So this is going to be collecting tissue fluid after it has exchanged. So after it has exchanged with the cells, it's going to be collecting that tissue fluid back. And then it will kind of go back in, kind of in vessels. And then it will also have nodes along the way, which will collect more and more of it. So there's also white blood cells inside of the nodes. So as it's kind of using the tissue fluid, if there's a lot of bacterial parts and so forth in the tissue fluid, it can help fight that off as well. Okay, so there is our little lymph capillary. So then that is going to connect with my venule, and my venule is going to then meet up with my veins. So if I go back to what the challenges are, so the challenge for my arteries is to withstand the high pressure. The challenge for my capillaries is to increase the speed of diffusion. And then the challenge for my veins is going to actually be to prevent backflow. So because they have actually lost pressure, so veins are returning to the heart. So first of all, they're more likely to go against gravity, not all of them. So we certainly have veins ahead of our body as well. But in the bottom half of our body, they're having to do that against gravity. But even the ones that have gravity working on their side, it's been longer since it's pumped. So this is going to be due to low pressure during return to the heart. So it's going to go back to the heart to be pumped, but in the meantime, it has lower pressure. So a couple things in place. So first of all, we don't need as thick of walls because it doesn't have that need to withstand pressure. It is going to have some protection, so it should be much harder to damage a vein than a capillary, which is important because they do have a large blood volume in them. So less pressure, but greater volume because they have a wider lumen. I should probably technically have drawn 3 layers, so close enough. So they have the same number of layers. The difference is just related to the size of them. So they still have the 3 layers, but the 3 layers are kind of all right on top of one another. And then the main thing that they have is the valves. So they are going to have the valves to prevent flowing the wrong direction. So the valves will allow it to flow in a single direction and then close behind it. So that will be really helpful. Okay, so that's everything, but I want to do one more. I forgot to leave a little bit of room for it, so I'm just going to offensively erase the vein really quickly, but leave it right here. Okay, last little thing that everybody needs to know about is occlusion. So that's kind of our last little point that we need to hit on. So an occlusion is a blockage in an artery, or can be in a vein, but they're most problematic when they are in our arteries, and the more important the artery, the more problematic. So let's look at the kind of what happens to make an occlusion occur. And you really just need to know kind of essentially 3 steps. So it's going to start with plaque forming in the vessel. As the plaque builds up, at some point it is going to potentially rupture. And so the plaque will start to kind of scatter. This rupturing is much more likely to happen if you have high blood pressure. So if you have hypertension, which is high blood pressure. So if hypertensive, it puts more pressure and then the plaque is more likely to break. If the rupture happens, the body, in its good intentions, tends to try to clot the plaque. So even though it's not actually like damage to the actual wall, it will often try to then repair that. And when the clot actually forms, then that will actually block blood flow. So that is going to reduce the flow. So that is what causes the blockage. And then this is most problematic in several different places, like the aorta would be crucial. The aorta is a bigger vessel, so it's a little bit less likely. The worst is the coronary arteries because they're not super thick like the aorta, but super important. So these tend to be the biggest problem in the coronary arteries. The coronary arteries are the arteries that branch off the aorta and return blood to the heart. So the aorta comes along and then it returns it to the heart muscle. So if you actually have a blockage in the coronary arteries, the muscle itself will not get what it needs and you will have a heart attack. Okay, so that's the last little thing that is everybody. So the next part on the heart itself is going to be HL only. I am going to erase just so I'm not having to write over on the side the whole time. But you guys are going to be in the second half of your page now. If you are SL, you do have one more thing that you need to know about, which is going to be to come back for a little bit of xylem. So when you see me starting to draw some xylem, return. So fast forward till you get to there. Okay, so when we talk about the circulatory system and the heart, we need to define this idea that it is in fact a double circulatory system. And if you recall, I said that this whole topic has a weird little but what about fish theme. So fish have a single circulatory system. So the double is distinct to mammals. And so the key is that they actually return blood to the heart before it goes out to the body. So returning oxygenated blood to the heart. Fish do not do that. They pick up the oxygen, from that same pump, deliver it to the body, and then it returns, so it's lower pressure. So that is our moment of what about the fishies for that. Okay, so there are two double circulatory systems, just refers to two systems. So we have the pulmonary circulatory system, and the pulmonary circulatory system is going to go between the heart and the lungs. And then we have the systemic system. And that is going to go between the lungs and, the heart and the body. You don't need to identify like which parts are which. So it wouldn't say like, what is the left atrium? If you know your tracing of blood, you could probably work it out anyway. All right, so we are going to draw a little heart. This can take up about, mine's very small. You probably could get away with taking up half of the remaining space. So let me talk you through how I'm going to draw a heart. I'm going to leave a little bit of space for the things. So I'm going to basically kind of draw it like I would if I was drawing a fun fictional heart. So I'm going to draw almost like a butterfly shape. These are going to represent, probably a little bit big, like so. So again, kind of like a butterfly. And so that is going to be my atria and ventricles. Then I'm going to go ahead and draw my vessels so I don't forget about them. So, I mean, I don't know, my atria are very big. It's fine. You get the idea. So, let's draw some vena cava. That's fine. Then this is going to go from here out. So, then this is going to come in. Sound effects. Then this one is going to go out and up. Okay, they overlap there. That like slide, if you can just leave it overlapping, or you can erase some of the overlapping because you're offended by it, and surely by now you're using Frixion pens. I'm not sponsored by them, but I should be. There you go. So there is my heart drawn. If you ever felt the need to draw a heart, isn't this an exciting tutorial on how to do that? Okay, so we're gonna label it and we're gonna put it in numerical order. It does say you have to be able to trace the unidirectional flow, so you can basically from the vena cava out the aorta. So you wouldn't be able to need to do it in that order. So first up we have the vena cava. So one vena cava. Perfect, so you do have a superior from your head, inferior from the bottom half of you, both delivering blood to the same place, which is going to be to the right atrium. 1 vena cava, 2 right atrium. In order to go between the right atrium and the left atrium, the right atrium and the right ventricle, we need to go via the tricuspid valve. And I'm writing on the heart. I did a bit of a combination of both. I had to do some arrows, so just do whatever works for you. So we're going to go vena cava to right atrium, down the tricuspid valve, into number 4, the right ventricle. The right ventricle is now going to make a pump. We'll go through the actual ventricular systole and all that next. Right now we're just doing the anatomy. Boom! Pump is going to pump and it's going to have to go through a valve. So anytime we are trying to move out, we want to ensure it doesn't come back in. So that is going to be the number 5 pulmonary valve. Remember, in the end, you're going to have 4, 4, 4. So if you're finishing off, double check. Do I have 4 vessels, 4 valves, and 4 chambers? If so, then you've got it. You also have 6 and 6. 6 before the lungs, 6 after. So those are just ways to check. Okay, so the pulmonary valve, and then it's going to come out here into the pulmonary arteries, or one big pulmonary artery that branches off into 2, which is the only deoxygenated artery in the body because it is going away from the heart but going to the lungs, so it does not yet have oxygen. Switching color. So I find red and blue as an easy cue. Do remember that your blood is never blue. It is slightly redder in the presence of oxygen, very slightly. But it still makes an effective memory strategy to separate it. So now we are going to come back in through the pulmonary veins. There are 2 of them because 1 comes from each lung. Going to come from the pulmonary veins into now the left atrium. Because it is going to go down into the ventricle and we don't want it to come back ever again. Well, not yet. We are going to go through a valve called the mitral valve or the bicuspid valve. It's going to then enter the left ventricle. The left ventricle is going to be super, super muscly. So I'm just going to make a little note that this is the thickest muscular wall because it has to deliver blood with enough force to make it all the way through the whole body, not just to the heart. So nice thick muscular wall goes boom. So then it's going to come up, and we definitely don't want it to come back because we just used all this muscle energy. So it's going to come here through 11, the aortic valve. And finally, out through number 12, the widowmaker itself, the aorta, the most important vessel in your body. If you have a problem in your aorta, it is very bad news because it is meeting the needs of the entire rest of the body. So again, if you were labeling that, you would want to go through and check you made it to 12, count your 4, 4, 4, count your 6 and 6, and you would know that you are good. 2 more things I want to label in here. First of all, you do need to know what is this area here. So that is the septum. So that is going to be the muscle that separates the 2 parts. So that's going to be important. And then the other thing is I just want to remind us what these valves are called because we're going to use their other terms in the next part. So the tricuspid, because it's between an atria and a ventricle, is an AV valve. And then the mitral is also an AV valve, which just means between atrioventricular, between an atria and a ventricle. The other 2 valves are between the ventricles and the actual big vessels of the body. So these are called semilunar valves. That term makes a little bit less sense to easily remember. But for the next section, we're not going to write over and over tricuspid plus mitral. We're just going to write AV valves. So just be clear on which is which would be helpful. Okay, so that's the structure of the heart. Again, I'm going to erase just so that I have a nice big space. The next thing, and hopefully you have a little smidget of space left because we're going to do the cardiac cycle. So your cardiac cycle is the events that occur in a single heartbeat. So every pump that your heart makes goes through these same steps. So I am not going to draw the heart over and over. I did that when we did it in class. So you can have a look at that. I'm going to draw one little butterfly heart over to the side just to label the different parts. But then we're just going to do it in steps. So we'll just do step by step. All right, so here's my butterfly heart. Beautiful, like so. Okay, so we just have that there so that there's a couple things that we're gonna label. So every cardiac cycle begins with the sinoatrial node. So here's gonna be my sinoatrial node. It's gonna be in the top of the right atrium. So that is gonna be SA node. Okay, so that is going to be the first step. The sinoatrial node is so special because it is the only set of neurons that can independently fire without your brain, which is why you can be brain dead but heart alive. So it is able to independently, without even your brain stem, maintain its actual heartbeat by sending its own action potentials. So that is the sinoatrial node. And it is going to, again, we're just going to say independently fires action potentials. And those action potentials are going to go to the atria, so to the walls of the atria. So it's sending kind of messages all the way into the atria and to the atrioventricular node, which is going to be the AV node. So I don't know why I wrote it in full one place and not the other, but it's fine. Same idea. So it's going to send action potentials to the atria and the AV node, and then it is going to pause at the AV node for 1.2 seconds. Which is going to be important. And so then it is going to pause for 1.2 seconds. So it's alerted the atria, and then it's sitting at the atrioventricular node. The atrioventricular node is going to have to fire to start the ventricles. So during this time, so during the pause, that is going to allow for the atria to contract and empty their contents. So atrial systole is going to happen here. So this happens during the pause. So the atria are going to contract. And they are going to send all the blood into the ventricles. During this time, the SL valves are going to start open, and then they're going to close once the ventricular pressure is higher than the atria. So AV valves will close once the ventricles are filled. So once the ventricles are full, that will create a pressure that will close those valves. So they're going to start open, but then they're going to close. Now, our time has passed, and now my 1.2 seconds is over, and so then the AV valve, I shouldn't have drawn them the same colors, but anyway, is going to start sending its messages. So next thing that is going to happen is now the AV node is going to fire to the ventricles. So it is going to let the ventricles know that it is time to contract, and it's going to be really important that during this time, my semilunar valves are going to be opening. Now that those semilunar valves are open and the ventricles have gotten the message, they are now going to contract, and that is going to cause ventricular systole. So now my ventricles are going to contract, and they are going to empty the blood out of the heart. And then we will note that once that is done and it is empty, the SL valves will close. So as soon as it has removed the blood, those SL valves will close. Once they close, that will trigger the opening of the AV valves, and now the heart is open to allow accumulation of blood again. So now the diastole is going to happen, and during diastole, the AV valves are open, so they are accumulating blood so that they have a good amount of force. So the AV valves are going to be open, so they are allowing blood into the atria as well as into the ventricles. So remember, during this time, we don't accumulate it only in the atria. So there's a little bit of that happening earlier when it's closed, but now they are going to be open, and that is going to be allowing the heart muscle to relax and the heart refills. So that is going to continue until the atria next get the message to contract again. Then they will push their blood into the ventricles, increasing its pressure, and it will go from there. Okay, so that is the steps. You would need to be able to remember the general idea. I think the most important thing is if you can't remember the valves, it's not the end of the world. The most important thing is that you know starts with the sinoatrial node, then the atria contract, then the ventricles contract, then relax. So even if you can just get kind of those steps and not worry too much about the valves, if you can, try to remember which valves, but if you remember what's going on, you should be able to remember which valves. So if you're like, the atria are pushing blood into the ventricles, you should be like, oh yeah, the valves would have to be open for that to happen. If you're like, the ventricles push blood out, you'd be like, oh yeah, the SL valves need to be open. So you should be able to work out who's open and then the other one's just shut. So I wouldn't worry terribly much about it, just focus on the events, push, push, relax, and you should be able to work out your valves from there. All right, last little thing is that you do need to be able to draw, and if you don't have much space, this can be very tiny, an electrocardiogram or EKG outcome. We don't call it an ECG because that is a brainwave. So that's why it turned into K. So you would need to be able to potentially draw this. They have asked that before, or label it. So we have straight, bump, down, up, down, back, bump, done. So memorize the shape. So then you need to also know what each thing represents. It's not overly complicated. First, you don't necessarily need to know why. So this is a great exercise in just memorizing it. So you don't need to count, for instance, but why does the diastole cause a bump? Why does it go below it? So just learn what each part is and your life will be happy. All right, so this is going to be atrial systole is the first change. That represents that firing. The big QRS. I don't know why it couldn't just all be called Q, but I didn't make these things up. So this is a ventricular systole. So that is all three parts. And then T represents diastole. There you go. Memorize the letters, the shape, and what each one represents, and you will be fine on that. Okay, SLs, welcome back. We are now going to do plants. Plants is a little bit of SL and HL. So we'll do the SL part first, which will be the xylem. And then we'll pick up the HL part, which will be the phloem. So 2 parts for that. All right. Okay. So now let's look at transport in plants. So I'm going to draw down kind of the center-ish. My transverse sections, so my tissue transverse sections for the stem and the root, because you have to know those, and then we'll kind of draw a line from the xylem and do it on this side and the phloem on this side. Okay, so this is everybody. This is going to be my stem. This is going to be my root. And the green is the epidermis for everybody. Which can also make cuticle, particularly in stems. All right, next thing is that you need to draw, we'll go ahead and draw the vascular bundles and then we'll fill in the other space. In terms of what you absolutely need to know, you just need to know the xylem and the phloem. So we will just draw those, and then SLs, you do need to know what they look like. You just don't have to know the phloem in terms of what it's doing. Phloem sits on the outside, and the vessels are smaller. So if you were drawing it, we would draw the smaller circles. Then the xylem sits on this side, and they're a little bit bigger. Doesn't have to be dramatically bigger. More, you would recognize it if you were shown it in a micrograph image, that the bigger one is the xylem. Remembering that together they're called a vascular bundle when we're looking at them together. So we can say that that is a vascular bundle. In the roots, they look a little bit different. So I'm going to do the xylem first in the roots. So it tends to exist in this cross pattern. And mind you, these are dicot stem and roots, which is a term you don't really have a lot of context for it at the moment because you have not learned dicots versus monocots. So just remember this is dicots, which is just a type of flowering plant. So then we draw kind of a center one, and this time the xylem is kind of all central, but we do want to draw slightly smaller circles for our phloem to show that. Okay, and then everything around between the epidermis and the vascular bundles is called cortex, ground tissue, but they're using the word cortex. And same, this is all cortex. There is another name for the inside, then it's called the pith. It is not just empty space, it is made up of a different type of tissue, but you're not required to know that one. So you just need to know cortex is the tissue between the epidermis and the actual vascular bundles. Okay, so then we're gonna say you have to know about each of these in more detail. So we'll go xylem on this side, and then we'll do phloem on this side, although we will have to draw xylem again because of their interaction. Okay, so all things on this side are HL only. And everything on this side is everybody. Okay, so I'm going to try to draw several things all on one diagram. So let's draw some soil. Here's some soil. Okay, so in the soil, I am going to draw a root cell. Coming off of that, I'm then going to draw my xylem. Like so. I might not be very good at drawing xylem. Flexion. All right, something like that. There's some xylem. And then we will draw, coming off of the xylem here, we will draw a leaf cell. And then in the leaf cell, we'll draw a stoma. So that's my stomata. Okay, so we need to know structural things about the xylem, and then we also need to know a little bit about the movement of water through it. So structural things, let's label them. So in the xylem, to make it extra strong, there is a polymer called lignin. So that is going to be the lignin polymer. You don't really have to know what it's made up of. And it is going to strengthen those already thick walls. So that's going to make them really thick, which is going to be good for pressure changes, because when they're full of water, they're going to have more pressure. When they're lighter in water, they're going to have less pressure. So it is going to provide kind of protection from the environment, but also kind of dealing with the pressure changes, which is really important. Okay, then we are going to have, let's make a note here that we have, I don't know where to put it, no cytoplasm. So that is actually allowing for kind of easy flow, so continuous flow. Similarly, you'll see that you have these broken down cell walls. So again, that allows for that continuous flow because it makes it a continuous column. So it's kind of one column rather than being interrupted by cell walls. And then we have the little things here, which are going to be the pits, and the pits are going to allow water in and out. And that's going to be really important for HLs to understand the movement of water between xylem and phloem. You need to have the pits for that. Okay, so that's the structural thing. Next thing we need to look at is all of the things that are pulling water. So water is coming out. Sorry, I should have drawn it like that way. So it's coming out of the actual stomata. So that is actually pulling water out of the leaves. And we've already talked about that, but in particular, we'll call that transpiration pull. So when we're talking about the force that transpiration pulls on water to move upwards, we don't just call it transpiration, but transpiration pull. So transpiration is the loss of the vapor. The pull is it's pulling all of the water up. So as that's pulling that out, it's pulling the next part up and so forth. So that contributes to it. We also have it moving in this direction. So the pushing of water from the ground up is also occurring. The term for that, which you actually only need to know if you're HL, is root pressure. Having said that, if you're SL, I would think it would just be worth learning the term root pressure than trying to describe the pressure exerted on water from the roots trying to move up. So root pressure is the water coming up, and that's because of kind of the full vacuoles. So the idea here is this is the positive pressure potential inside the vacuole is exerting a pressure that is going to pull the water up because of that positive pressure. That positive pressure is built up through osmosis. So osmosis moves the water into the root. Root pressure pulls it up into the xylem. Let's remember that if you don't have any transpiration pull, it's harder to move because water does accumulate in the roots a bit more. And so sometimes in order to do this, that is going to be after the active transport of minerals. So we're going to actively transport the minerals into the roots. That pulls the water by osmosis. That is technically HL only, but I think it's good for everyone to know. Last bit, which is everybody, is then the movement from here to here is called capillary action. So that is going to be that upwards movement in a thin tube. So capillary action is kind of the middle part. So osmosis, root pressure, capillary action, transpiration pull. So capillary action is that movement upwards. It is influenced by both root pressure and transpiration pull, but this is referring to the specific movement in the xylem itself. And remember that that is due to two forces. That is due to cohesion between water molecules. That is going to cause it to remain a continuous flow, and it is also caused by adhesion to the cellulose walls, which is going to essentially pull it up into each new cell. Not doing a ton on that because we covered it quite a bit in the water unit. So hopefully you have that elsewhere, but that is kind of the last step in that part there. All right, SLs, you're done because the last part on muscle and movement is HL only. So there you go. Go study. Be fabulous. HLs, you got a little bit more to go. Okay, so let's move on to phloem. So for phloem, we need to know about translocation. So translocation, if you recall, is the movement of sap. So in phloem, we have translocation instead of transpiration. Translocation is the bidirectional movement of sap or sucrose. Sap is made up of sucrose. We'll cover sucrose later. Okay, so we're going to draw very similar to what we drew in class. I don't think there's really any other way to draw it. We're just going to do a little bit simpler in terms of not writing so many steps. So we need to draw our little xylem and phloem. So since we drew xylem in blue there, we're just going to draw a simpler version of it over here. Okay. It is thicker, remember. I haven't drawn it a ton thicker because of spacing reasons. I need to have room for all my phloem fun. Okay, when we draw phloem, we need to draw two things. So again, it should be a little bit thinner. It does not have the totally empty walls like xylem, so you should have the sieve plates in between. So those are going to be the sieve plates, like so. Then they need companion cells because phloem has to use some active transport. So xylem, the active transport is only happening here in the root. Otherwise, xylem is all related to passive processes. So they don't actually need any ATP for anything they're doing. Phloem does because it is going to move things against their concentration gradient at a couple places. So as a result, they do need some actual living organelles that are going to help make them energy. So give them each a companion cell. And then make sure they have some plasma desmata. And they should also have plasma desmata here so that they can also move, which in theory is really going through the pits. So actually, sorry, the xylem wall does not have the gap. It has to go through the pit into the plasma desmata. My bad. All right, and then the last one needs one as well. And these again are living cells. So they should have some nuclei, some mitochondria, and so forth. That's a giant mitochondria, and so forth. Okay, so those are going to be my companion cells. So this is a sieve tube element or cell. Debatable whether it is a cell due to its lack of living things. Similarly, we don't say xylem cells, we just tend to say xylem vessels. This is a living cell called the companion cell. And the cytoplasmic connections between them are called plasmodesmata. So plasmodesmata in between them, and you do note I did draw them on both sides because that's going to be helpful for the water movement. Okay, it is bidirectional, but we are going to draw it a one-directional just for ease. So here we are going to draw a source. And the source is generally a leaf cell. Some stems also do photosynthesis. So in some cases, it could be from the stem itself, like a cactus doesn't have leaves, so it is the job of the stem. But in general, we will think of it as a leaf. Then we will draw down here my storage, which is going to be my sink. And this is going to be a root cell, but let's make a note, it could also be fruit. So that is why it can also be above, and sometimes they will release even at actual stem further like just stem ground tissue cells to meet the glucose needs of the stem. So a sink is generally more of a storage. So if we're really sinking it, it's generally the roots or the fruits, but it can leave at kind of companion cells at any place if they need the glucose. Okay, so let's look at what's going to happen. So I'm just going to do a what is happening at the source. And then we're going to look at what move the camera. There we go. And then we're going to look at what's going to then happen at the sink. So again, I'm not going to do it step by step. You have those in your notes if you want to go back to it. We're just going to do the main events. So the main event is going to happen at the source is that we need to load sucrose. So sucrose is what we are trying to transport. That is going to make up the sap. So we're going to load the sucrose from the source to the sieve. That is going to be active, then passive. So we are going to actively transport it from this leaf cell into the companion cell. So that step there is requiring ATP. Then it is going to slip through the plasmodesmata passively into the actual sieve cell. So active, then passive. Okay, when that happens, this is now building up a higher solute concentration. This high solute concentration is going to mean that this area here is, as a result, going to have a very low water potential. The low water potential means that as the water is moving up, it is going to move through. So I probably should try to draw it more accurately than that, sorry. So what it's going to do is it's going to actually come out of the pit and into the plasmodesmata. So water is, when it's traveling up, it's going to leave through the pit, enter the plasmodesmata, and enter the cell. So now it is going to transfer water from the xylem, and hopefully you remember, but if you want a label to remind yourself that this is the xylem, we can show the water is going to go there. The result of that is that this is creating a pressure, so it is going to move due to hydrostatic pressure. So it's moving away from that hydrostatic pressure, which is due to the water accumulating there. And also because the water potential will go up in that area. So kind of both. So from the pressure, but also the accumulation of the water and the water potential change. So as you start adding all this water, it is going to dissolve into sap. So this is now sap because it's the sucrose plus the water, and it would be moving away. And remember, it will also move in this direction as well. So it is just moving away from where it was loaded. Okay, then it is going to arrive at the sink, and at the sink we are now going to unload sucrose. So we're going to remove the sucrose out of it, and now we're going to unload it from sieve to sink, and that is actually this time going to be passive to active. So this time the sucrose can go through the plasmodesmata, like so, but then they will require active transport to go out into the sink. Once the sucrose is gone, what we are left with is just water, and that water will now return to the xylem because the water potential now is greater in the phloem than in the xylem. And again, I probably didn't draw it quite accurately because it would need to go out of plasmodesmata into a pit. So that's just the general idea of it. Okay, so that is all things transport. Last bit will just be muscle and motility for HL only. Okay, lucky last. So this is an all HL topic, so HL only. It's also not super big compared to the last ones because it's only HL. So we can probably, I'll still do a page, but they're not quite as crammed in. So that's nice. We're going to start big and go small. So we're going to start with why move, big picture, how do our muscles move our bones? Then we're going to go a little bit smaller into looking at motor units, then sarcomeres, then the sliding filaments. So that is our plan. So we're going to go why, how, when. So we're going to start with why do organisms move? So why is it beneficial for organisms to have the ability to move? And we are talking here about locomotion, not sessile organisms who move part of their body. There are 4 reasons why motility is an adaptation. So one is it allows for foraging of food, which is important if you are a heterotroph who cannot make your own food. Then it allows for escaping from danger, which is both predators, but also could be environmental dangers as well, which means less needing spines and other things like that because of the ability to run from trouble. The next one is more ability to search for mates, which is really important because it cuts down on inbreeding and increases genetic diversity. And last one will be migration, the ability to seek out your zone of tolerance and therefore be able to survive better by changing environments as needed. So those are the official 4 reasons. Then next we're going to look at how. I'm going to leave a little bit here to the side. So we're going to come back in a second to when. Okay, so over here for how. So how does that actually happen? How do we actually create movement? So for that, you need to know about 3 things. So it happens because muscles lift bones at joints. So all 3 of those we need to be kind of clear on. So the role of all of those things. So those are all really important. I'm going to draw the little lever model just as a reminder that that is kind of an analogy for how this happens. So there's my bone, and then my muscle is going to be the effort that is going to pull it up. And that's going to come from the muscle that enables us to lift any sort of load that we might have, or just even the load of gravity pulling us down. But we can only lift it on the fulcrum, which is the joint. So the joint creates the fulcrum on which the bone as a lever can move. So again, you don't need to know the different types. That's just that analogy. So making sure that you could kind of connect the different parts to it. One thing quickly when we say bones, that is a specific term for those of us who have endoskeletons. So endoskeletons are bones underneath other tissue. Reminder that other organisms work very similarly. Exoskeletons also move at joints, and the muscle also moves them, but instead of having bones, they have chitin plates. Almost all of the stuff that we have talked about has related to endoskeletons, so that's just a little tiny reminder. And then the other little small thing, remember, is the what about the fish? So we will kind of put somewhere a little fish maybe, but just remind you that all of these kind of muscles and how they work. We're mainly looking at it as if we were in air, but there are more challenges if they were swimming. So they have to have extra adaptations if that was the case. So just a reminder about that. I'm just not going to spend a lot of time on it because I think hopefully you could remember streamline shape, fluke, blowhole, the kind of basics of that. Okay, so let's do a little bit on the joints then. So we're going to use the joints to kind of cover a couple different things. So we are going to draw 2 joints. And from those 2 joints, we can kind of label our key parts. So we're going to draw a hinge joint. And we're going to draw a ball and socket joint. And just a reminder that your joints determine your range of motion. So the hinge is going to be the limited range of motion. So it just does the extend and flex. So flex and extend, while the ball and socket is going to have a wide range because it is able to also do flex and extend, but it can also do abduct, adduct. Can't really see my legs doing this. If I do this, abduct, adduct, and then it can also rotate. So your shoulder is also one. So I can go extend, I can go out and in, and I can go rotate. So more range of motion for that. But otherwise we are going to use these ones just to show a couple things. So let's draw your arm as your hinge, although your knee would also work. So this is going to be your stationary bone. This is going to be your bone that moves. You don't need to know the names of these bones in this case, but this is the one that is actually going to act as the lever, which will be your radius, ulna, one of those arm bones. This will be your humerus. And so that is going to be there. Let's note that you're going to have muscles. And I do want to actually draw both muscles. And again, I haven't drawn this with super accuracy because I probably should have a radius and an ulna. So this one should attach to the ulna and this one should attach, I'm just going to, levers. There. There we go. So you have 2 pairs. So remember, we always have antagonistic pairs. So that's what we're showing here, is that the muscles are always in antagonistic pairs. So this is, of course, the bicep, and this is going to be the tricep. So bicep and tricep. Okay, so now we need to connect our muscles to the bones. So we're going to connect to the stationary bone, and then I'm going to connect to the lever that I plan on using. I'm going to connect to the stationary supportive bone, and then I'm going to connect to the bone that I plan on moving. So those are tendons, and tendons are going to connect muscle to bone. Okay, so there's my tendons. I also need to connect my bones to each other. That's going to be done with ligaments. So ligaments are going to be very elastic, and they're going to attach bone to bone at the joints. Amazing, and then those are the main things I want to label there. I will go ahead and draw some synovial fluid because I'm going to use it on the next one as well. And then inside is going to be my synovial fluid. Okay, let's look over here at then drawing the hip joint. This is the named joint that you could have to know. So in this case, we will actually name the bones. So this is going to be your pelvis. And this is going to be, sorry, it's been a long video. Pelvis, and this is going to be your femur. Okay, so those are the only 2 named bones that you could have to name. So just be aware of that. So that the bones don't actually grind on each other, and this would be here as well. They're going to have cartilage on the outside. So at the very edges of them, so that's going to be cartilage. And that is going to absorb shock and prevent kind of bone to bone contact. So they rub against the cartilage rather than on the bone itself. So that's going to be important. We need to draw our ligaments again. Okay, and then inside we are going to have this synovial fluid. I'm going to label it in another color because you're not going to be able to see it in the yellow. I probably should draw it in brown. Okay, so that's my synovial fluid. And that is going to be inside the joint capsule. So that's going to be inside kind of the joint that's made by the ligaments. And that is going to be a lubricating fluid. And it's going to reduce friction. Okay, so those are essentially the joints that you would need to know, is just and then the structures related to those joints. The other thing I'm just going to add so we don't forget it here is, I'm just going to make a little note that there are extra adaptations for swimming. Sorry, hopefully you have room for that. So just a reminder to go back over those quickly if you don't know them. Basically streamlining and fluke. I'm just going to add those words in as little reminders. Streamlining, fluke. Okay, so you can quickly look at that, but if you're watching this for your exam, I don't want you to forget about that dot point in case, heaven forbid, you have some sort of 7-mark question about swimming adaptations or all the little fish parts in this topic. Okay, so that's how. Let's look at a when. So when does a muscle know to move a bone? So that is going to relate to when it gets the message from a motor neuron. So it is going to move when motor neurons release a neurotransmitter. We might as well know that it is acetylcholine. And they're going to do so at the neuromuscular junction. So by the time you're revising for your exam, we will have done more on neurons, so that will be helpful. But for right now, we haven't really done a ton of that yet. But we should know enough to know that they release neurotransmitters, etc. Okay, so this is going to be the cluster of muscle fibers that are going to be controlled by it. So each of these is a muscle fiber, which is another name for one long muscle cell. Okay, so muscle fibers, one long muscle cell. And then here's the motor neuron. And this one motor neuron is going to release onto that. And again, the actual neuromuscular junction is basically the little space where it's going to release its acetylcholine. So there's the acetylcholine, and that is going to be received by the surface of that. This together is called one motor unit. So that is a single motor unit. That is kind of one motor neuron delivering acetylcholine to one set of muscle cells that are all going to move together. Okay, so that is kind of when the muscles will move. So now I'm going to erase all of that. So we have come down to the second half of the page, and we're going to look at kind of the zoomed in version. So this is all still kind of the zoomed out version. Now we're going to look at what's going on in single sarcomeres. Erase. sarcomeres. So let's make sure we know where a sarcomere is in terms of on a muscle cell. So we have one muscle fiber. That muscle fiber is going to be a single cell, but it is multinucleate. So it's going to have nuclei. They generally sit on the outside. It's also going to have lots of mitochondria, an endoplasmic reticulum, all of those things. The reason why that is all sitting on the outside is because inside it is filled. Sorry, I drew a very small, unnecessarily small muscle fiber. So now I have no room for myofibrils. Okay, let's try again. Give it a little bit more space. So the inside of it is filled with these long fibers called myofibrils. So each one of these is the inside of one muscle cell, and this is called a myofibril. And then each myofibril is made up of a bunch of sarcomeres. This is what creates the striations. So prior to learning about this, we had kind of said that skeletal muscle is striated, but we had not actually accounted for what those striations are. So the striations are going to be where the myosin is, because it's going to cause a darkening pattern. So there's the myofibril, and it is made up of sarcomeres. So let's then look at each sarcomere. So this would be essentially one sarcomere. Okay, so we're going to label a sarcomere when it's contracted and relaxed. Then we'll go into how it actually does that. So when we draw a sarcomere, we draw Z line to Z line or Z disc to Z disc. I want to try to clarify that when it's contracted, they're quite far apart. When it's relaxed, they're quite close together. So try and draw a difference there as best as you can. The more distinct, the better. Okay, so in its relaxed form, the actin, which is the thin filaments, are fairly far away from each other. So we should have a clear kind of light band where there is no actin. Oh, sorry, it will become the dark band, but a clear space. In between there is the myosin. The myosin should be drawn thicker, so if you ever draw a sarcomere, make sure to draw that as the thicker band, like that. So if you were looking at it on an actual micrograph, this would appear dark. And this area would appear light on both sides. Okay, other little component that we need to add is the titin to it. So when we draw the titin here, the titin is going to be stretch. So that's going to be the stretched titin. Okay, so there is my sarcomere in its relaxed form. To go from this to this, it is going to contract. When it contracts, and we will go into this, how this happens in a minute. When that happens, the actin is going to be pulled in. So you should show very little space between your actins. Like that. So now your actin is pulled together, so your Z lines are closer. So your Z lines are less far apart because they got pulled in. the myosin hasn't actually changed. It's just the actin that changed locations. Like so, and then in this case, these are going to be coiled. So they are going to coil up when this happens. And when they're coiled, they're also storing some potential energy that's going to be important. Okay, let's make sure that we label the two types. So that's going to be my myosin, and that is going to be my actin. So when it is contracted, we see the shorter distance between the Z lines. Make sure you can see that. And what we're really seeing is that the actin pulled in, which we'll look at how that happens next. So the actin has pulled in. From contraction, it can go back to relaxation. But in order to fully relax, it does need to re-stretch the tighten. So in order to get it back into its fully relaxed form, it will need to use energy to actually relax the tighten, which is why muscles need antagonistic pairs. So this is done by the antagonistic pair. So we cannot actually relax a muscle without another muscle helping. So if you think about it, if you pull your arm up with your bicep, even if you relax your bicep, it's like you're not using it anymore, it can't totally relax unless you pull with your tricep. So once we've pulled, we actually require the other muscle to return it. So if I want to go back to relaxing the tighten, my tricep will work. It may not be a lot of movement, it may just be a little bit of the stored energy in the springs, but I will need something to kind of pull it back out. So springs in, and then in order to pull it back out and get that stored energy back in the springs, I do need the antagonistic pair. So that's kind of a reminder that we have antagonistic pairs, which we mentioned before, but this is the why. And then a reminder, they do like the actual example. So when we talk about those, we talked about this under our gas exchange, but remember that the external and internal intercostals are an example. So in particular, we use the internal intercostals, and so essentially these contract, and when these contract, these relax and stretch, and that is actually what causes the expansion of the ribs, is the actual stretching of the intercostals. Okay, so that's just a reminder. I think that's all the little bits and pieces. If you were asked about an antagonistic pair, that is the one they've named. You could use bicep and tricep if it gives you the choice, but if they ask specifically for the external and intercostal, again, go back to your gas exchange stuff because it fits in nicely with there. Okay, so last thing we're gonna do is, how does this actually happen? So this actin is pulled in. That is the sliding filament theory. So now we want to say, how does that actually happen? So we are going to go into the sliding filament model theory, whatever it is. So I'm not going to draw it as a circle. I'm going to draw it as 4 separate steps, and then it will kind of restart here, but just because it's a little bit easier to draw it linearly than having to draw it in a circular motion. So we're gonna draw it that way. So this is the sliding filament theory. Okay, So the sliding filament theory is how does actin get pulled in? So we say the sliding filament theory, it is basically how myosin pulls in actin in order for muscles to contract. So myosin pulls in actin for muscle contraction to shorten the sarcomere for muscle contraction. So I'm going to number the steps, but it's a little, you could in theory, because it's a cycle, there's not actually a number, if that makes sense. So I've gone back and forth whether to even remove the number, because some will start at here and call this 1, 2, 3, 4. I'm just doing it a slightly different way. I'm going to actually start with the ATP coming in. So I'm going to put the numbers, but the numbers are not, this is not, it is a cycle. You could start at any number and go from there. Okay, so I'm going to, like I said, I'm going to start. The first thing that's going to happen is we're going to actually take in the ATP. That's actually going to allow my myosin to detach from the last contraction. So here's my myosin. And again, I'm just going to draw this a little bit more 2D because we're just trying to make it a little bit simpler. There we go. And this had previously been attached to my actin. So it is going, oh, sorry, I should draw it in pink because that's what I drew before. Like so. And so the way that I was able to detach it is because ATP bound to it. So the addition of ATP enabled it to detach. So that is going to be my first step, is that the binding of ATP causes the myosin to detach from the actin. Okay, so this is I'm gonna start with the muscle relaxing and then show what happens when it contracts. So step 1 is detachment. The next step is gonna be, I'm gonna call it relaxed but ready. So the next step is gonna be that it is relaxed. So it is not currently contracting, but it is ready to go. The other thing you could call this is ATP hydrolysis, which we'll label it below. So now what's going to happen is, with the ATP, it is going to go ahead and hydrolysize that ATP, and in doing so, it's going to allow the myosin heads to change shape. So normally straight up, when they break down the ATP, they're able to move forward. So that is going to happen at this step here. So now instead of having a TP attached to them, they have ADP and a loose phosphate. Again, they are not currently attached. The reason they're actually not currently attached is because of a block. So the actual binding sites are blocked. They're actually blocked by trypsomycin. You don't have to know these terms anymore. So they're blocked, and then you're going to need calcium to unblock them. This is important because what it means is your muscles are not acting independently, so they are waiting on the message, the calcium, to come through and move the actual blockages to encourage the muscle relaxation. So the shape is appropriate, but they are actually blocked, not actually binding at this point. So this is going to be ATP hydrolysis, which means they're breaking apart the ATP. That is going to change the shape of the myosin heads. but it is currently not binding yet. So it could bind when sites are available. So when those actin sites become free, it is ready to go. So what is actually happening right here during this stage? This is all part of muscle relaxation. So at this stage, the muscle is not actually contracting. So the actin has not been pulled in. So right now it is in a state of muscle relaxation. Okay, so detaches, gets ready. Now, this is going to be with the addition of basically a message or a firing. So calcium is going to come along to unblock the sites. You don't actually have to know that it's calcium that does that. You're fine just to know the sites magically become open. So these, probably the next two are the titles you definitely need to know. So now once these become available, they are going to form what's called cross bridges. And you do need that term. So what has happened now is that there has been a change and my actin sites are now, make sure I have room for the last one, I should be fine. So they're still like this, but the binding site has become available, and so they are going to actually move up and bind to it. So they are actually going to bind to the actin. They still have right now ADP plus P. Like that. Okay? So that is the next thing that happens, is that when available, so when the actin sites become unblocked, the myosin heads will attach. Okay, now it is ready to then go, so now we need to move it. So then our last step needs to be to move it over. So this is called the power stroke. So what is basically going to happen is it is going to now push back in. So it is going to, thank you, push back in. So it is going to move. So here is, they are going to go back to upright, like so. And in order to do that, they're releasing the ADP and P that was holding it. Okay, so release the ADP and P, and then they go boom, bounce right back up, and then they are still connected. But when they did that, they went, so now this got pulled in. So in moving, this got pulled down. So when they flipped back up, they moved it along. That is the power stroke. So it's when the heads bounce back up, they are going to then move the actin in. So the removal of the ADP and P brings the heads back to their original shape. And then in that process, they pull the actin in. So that is pulling the actin in. So we said this is all things muscle relaxation. This is muscle contraction. So what we see where we left off is that it is still attached. So then the next step is going to be to then add more ATP and detach it. So you can really put it at any place. I could have started it here, but I liked to show that that was kind of normal position. this was what you had to hydrolyze the ATP to get to, then bind, then bounce back, but then you will need the ATP to release so that you can start again. So then it will restart the cycle when you add ATP, and that allows it to detach, which will be necessary for it to attach again. So again, the numbers are arbitrary, the sequence is what matters. So detach, move, cross bridge, power stroke, however I'm drawing it, cross bridge, power stroke, and then start again. So make sure you could account for kind of the overall ordering of those steps. All right, awesome job, guys. Good job revising all this. Good luck for your tests and exams. Bye.